Hello viewers, in this session I will be discussing about placenta, its physiology, classification and functions. I wish to dedicate this PowerPoint presentation to my teacher at Guwahati University, Professor Jogen Chandra Kolita, who is a constant source of inspiration and encouragement in my life. Before I go into detailed discussion of this topic, I would like to mention here that this video is prepared as part of the laudable initiative of Zoological Society of Assam to teach the students of life sciences during this critical hour caused by coronavirus outbreak. After going through this PowerPoint presentation, you will be able to know about the definition of placenta, implantation of the blastocyst into the uterine wall, developmental and physiologic anatomy of the placenta, classification of placenta in different mammals, and functions of placenta. Now let me define what is placenta. The placenta is defined as a temporary organ or connection between maternal and fetal tissues via the umbilical cord that facilitates the nutrients and gas exchange between maternal and fetal compartments. Kent in 1987 stated that the term placenta in its broadest sense refers to any region in a viviparous organisms where maternal and embryonic tissues of any kind are closely opposed and which serves as a site for physiological exchange between parent and embryo. The word placenta comes from the Greek word for a type of cake or flat slab-like in reference to its round and flat appearance in humans. The mode of formation and fusion of the placenta to the uterine wall is called placentation. The phenomenon of placentation is invariably related with the viviparity of the organisms. In all viviparous animals, the development of the young one takes place inside the uterus of the mother, and the amount of yolk of the egg does not remain sufficient for the development of an embryo. Therefore, the developing embryo has to depend on the mother for its nourishment, oxygen supply, and other physiological assignments. The placenta begins to develop after implantation of the blastocyst into the maternal endometrium. After the process of cleavage and morula, the outer layer of the blastocyst becomes the trophoblast, which forms the outer layer of the placenta. And this outer layer is divided into two further layers. The underlying one is called cytotrophoblast layer and the overlying one is called syncytotrophoblast layer. Now defining the structure of the placenta including its umbilical cord. At full term, the placenta is a discoidal structure. It is about 22 cm in diameter, 2.5 cm in thickness. It weighs about 500 to 600 gram and dark reddish blue in color. Of course, under microscopic examination, it appears pearl white. When the placenta is viewed from the maternal side, you will see about 15 to 20 slightly bulging areas there, and these are known as cotyledons, which is covered by a thin layer of decidua basalis, and these are clearly recognizable here. Grooves between the cotyledons are formed by decidual septa. On the other hand, the fetal surface of the placenta is covered entirely by the chorionic plate. A number of large arteries and veins and chorionic vessels converge toward the umbilical cord. The chorion in turn is covered by the amnion. Attachment of the umbilical cord is usually eccentric and occasionally even marginal. Rarely it inserts into the chorionic membranes outside the placenta. Such condition is known as filamentous insertion of the umbilical cord. Umbilical cord is the anatomic tubular structure that physically connects the developing intrauterine fetus to the placenta, which is anchored to the maternal uterine wall. The attachment site of the umbilical cord to the fetus is called umbilicus or navel. At full term, an umbilical cord is about half a meter long. Usually, umbilical cord coiled in a counterclockwise direction, approximately 0.1 to 0.2 coils per centimeter of its length. Insertion of umbilical cord on the placenta is normally centrally located during the mid-gestational period, but may become more eccentric as gestation proceeds. At birth, the umbilical cord is cut and the leftover bit becomes the belly button. Cord blood contains all the normal elements of blood but also rich in stem cells. Cord blood stem cells can be used to treat over 80 different diseases. Umbilical cord allows the baby to move around safely. Now let me give you some insights into the early stages of placenta formation. Before placenta formation, cleavage occurs, and the cleavage is a series of mitotic divisions of the zygote. The process of cleavage forms a blastula consisting of cells called blastomeres. A cluster of blastomeres, which is 16 to 32 blastomeres, forms a morula. Blastocyst formation involves fluid secreted within the morula that forms the blastocyst cavity. The conceptus is now called a blastocyst. The blastocyst is usually implanted within the posterior superior wall of the uterus by day 7 after fertilization. Implantation occurs in the functional layer of the endometrium during the progestational or secretory phase of the menstrual cycle. Initially, the blastocyst adheres to the endometrium and the outer trophectoderm layer differentiates into syncytiotrophoblasts and cytiotrophoblasts. The syncytiotrophoblasts erode into the endometrial glands and blood vessels, now establishing a lacunar network here. Cytiotrophoblasts form a stem cell column for villus development. In this diagram, you can see that at approximately day 9 after fertilization, the blastocyst implants into the endometrium and the formation of extensive syncytiotrophoblasts at the embryonic fold is initiated, and it is the start of invasion of endometrial glands by these syncytiotrophoblasts and the formation of lacunae in this area. As the chorionic sac grows, the villi associated with the residua basalis rapidly increases in number, developing a brachy area called the villus chorion or chorion frondosum, which later evolves and forms the placenta. The intervillous blood flow is established only after the 12th week of gestation, and a slow flow, low impedance uteroplacental circulation is established. Now let me define the later events after day 9 of fertilization as well as after establishment of the fetoplacental circulation up to day 20. Here in this diagram, you can clearly see that lacunae are now well developed within the syncytial trophoblast 8 days after fertilization, and this lacunae facilitates the exchange of nutrients and other materials between the fetal and maternal blood circulation. Below, after 12 to 15 days of fertilization, the yolk sac is well developed and the chorion is delineated, and now the extraembryonic xylem is called chorionic cavity, and the conceptus is now suspended by the connecting stalk within this chorionic cavity. And after 20 days of fertilization, three germ layers are developed. They are endoderm, which is the innermost layer, the ectoderm, which is outermost layer, and the middle one is mesoderm, which is middle layer. These three layers gives rise to certain tissue types in the body. For example, endoderm gives rise to lung, liver, pancreas, intestine, stomach, colon, upper urogenital tract, and female vagina, whereas ectoderm forms certain outer linings of the body like outermost layer of the skin and hair. It is also a precursor to mammary glands, central and peripheral nervous system. And the mesoderm gives rise to dermis of the skin, heart, muscle system, bone and bone marrow. Now, in a later stage of development of this placenta, the primary chorionic villus continues to grow with the proliferation of the cytotrophoblastic cells. However, mesenchyme from the extraembryonic xylem invades the intervillus space, which forms the secondary chorionic villi. Eventually, these mesenchymal cells form fetal capillaries, and at this time the villus is known as a tertiary chorionic villi. The lacunae also enlarges by emerging with one another in this area, and the extraembryonic xylem later on pinch off as a exocoelomic cyst. Okay, the entire process of placenta formation may be discussed in a sequential order. First, the blastocyst is implanted on the day 6 after fertilization. The trophoblast cells of the blastocyst now interact with the endometrial decidual epithelium. On the 8th day of development, trophoblasts differentiated into syncytiotrophoblasts. On day 9, lacunae or spaces are formed within the syncytiotrophoblast. The syncytiotrophoblast erodes the maternal tissues, allowing maternal blood from uterine spiral arteries to enter these lacunar networks. Thus, early uteroplacental circulation is established by the end of week 2. Meanwhile, the cytotrophoblast begins to form primary chorionic villi or finger-like projections which penetrate and expand into the surrounding syncytiotrophoblast. In the third week, extraembryonic mesoderm grows into these villi, forming a core of loose connective tissue, at which point these structures are called secondary chorionic villi. By the end of the third week, embryonic vessels begin to form tertiary chorionic villi. The cytotrophoblast cells from the tertiary villi grow towards the decidua basalis and spread across it to form cytotrophoblastic cell. The villi that are connected to the decidua basilis through the cytotrophoblast cells are known as anchoring villi. Now, uteroplacental circulation is established after 2 weeks. By the 4th month, the placenta has two compartments, that is, the maternal portion called decidua basilis and fetal portion called chorion frondosum. During the 4th and 5th month, the decidua form the septa and the septa divides the placenta into compartments called cotyledons. The entire process of placentation is completed by the week 14 to 16. As already I have mentioned that by the beginning of the 4th month, the placenta has two components, a fetal portion called chorion frondosum and a maternal portion called decidua basilis. Here in this diagram, this is chorion frondosum, which is the part of the fetal placenta, and this is decidua basilis, which is the part of the maternal placenta. On its maternal side, the placenta is bordered by the decidua basilis, of which the decidual plate is most intimately incorporated into the placenta. In the junctional zone between decidua basilis and chorion frondosum, trophoblast and decidual cells are intermingled. This zone is characterized by decidual and syncytial giant cells, which is rich in amorphous extracellular material. By this time, most cytotrophoblast cells have degenerated. Between the chorionic and decidual plates are the intervillous space, which are filled with maternal blood. The villus trees grow into the intervillous blood legs from the chorion frondosum. During the 4th and 5th month, the decidua forms a number of decidual septa which projects into the intervillous space but do not reach the chorionic plate, and these septa have a core of maternal tissue, but their surface is covered by a layer of syncytial cells, so that all times a syncytial layer separates maternal blood in intervillous legs from fetal tissue of the villi. As a result of the continuous growth of the fetus and expansion of the uterus, the placenta also enlarges. Its increase in surface area roughly parallel that of the expanding uterus, and throughout the pregnancy it covers approximately 15% to 30% of the internal surface of. Now the development of the blood circulation to the placenta. From this diagram one can easily understood that how fetal villi or chorionic villi protrudes into the intervillous space, and it resembles a thick forest of trees arising from the chorionic plate, which is the analog of the soil from which trees sprout. Maternal spiral arteries undergo remodeling to produce low resistance and high blood flow conditions in order to meet the demands of the developing fetus. Cytotrophoblast cells invade the maternal spiral arteries and replace maternal endothelium. They undergo an epithelial to endothelial differentiation. This increases the diameter and reduces the resistance of the vessels. The maternal arterial blood is discharged from approximately 120 spiral arteries. These arteries have multiple openings, but not all the openings need be open at the same time. Blood enters in pulsatile spurts through the wall of the uterus and moves in discrete streams into the intervillar space. Small lacs of blood near the chorionic plate dissipate the force of the arterial spurts and reduce blood velocity. The maternal blood spreads laterally over the closely packed villi. Blood flows slowly, which allows adequate time for exchange. The principal factors that regulate the flow of maternal blood in the intervillar space are maternal arterial blood pressure, intrauterine pressure, and the pattern of uterine contraction. However, the fetal blood originates from two umbilical arteries. Unlike systemic arteries, after birth, umbilical arteries carry deoxygenated blood. As these umbilical arteries approach the placenta, they branch repeatedly beneath the amnion and penetrate the chorionic plate, and then branch again within the chorionic villi, thereby forming a capillary network. Blood that has obtained a significantly higher oxygen and nutrient content now returns to the fetus from the placenta through the single umbilical vein. Exchange of substances from one bloodstream to the other takes place by various transfer mechanisms such as diffusion, active transport, pinocytosis, and leakage. Leakage occurs by breakage of placental membrane. Now let me define the basis of classification of placenta in different mammals. The true placenta of the uterine mammals is a marvel of biologic engineering. Though the placentas of all uterine mammals show common structural and functional similarities, there being striking differences among species in gross and microscopic structure. Two characteristics, however, are particularly important, which form the basis of placental types. The first one is the gross shape of the placenta and the distribution of contact sites between the fetal membrane and the endometrium. And the second one is the number of layers of tissues exist between maternal and fetal vascular system. Based on the pattern of implantation, placenta may be classified into three different types. They are superficial or central placenta, eccentric placenta, and interstitial placenta. The effect of implantation in all cases is to obtain very close apposition between embryonic and maternal tissues. There are, however, substantial differences among species in the process of implantation, particularly with regard to its invasiveness or how much the embryo erodes into maternal tissue. In species like horses and pigs, attachment and implantation are essentially equivalent. In contrast, implantation in humans involves the embryo eroding deeply into the substance of the uterus. Many years ago, three fundamental patterns of implantation therefore were described based on the position of the blastocyst it assumes in the uterus. Now let me define how these three types of placenta are differ from each other based on the pattern of implantation. Here in superficial or central placenta, the blastocyst becomes attached to the surface of the uterine epithelium and thus lies in the cavity of the uterus, or it may remain in the center of the uterus. This type of placenta are found in pig, dog, and monkey. In eccentric type of placenta, the blastocyst lies for a time in a fold which loses off from the main uterine cavity. Such type of placenta are found in beaver, rat, mice, and squirrel. And in interstitial type of placenta, where an extremely intimate contact exists between fetal and maternal tissues. Therefore, the blastocyst penetrates deep into the uterine wall and the development of the embryo takes place inside the uterine wall. Such type of placenta are found in hedgehog, guinea pig, some bats, and ape. A mammalian placenta typically is a structure produced by the fusion of the extraembryonic membranes and the maternal single component, the endometrium. Therefore, based on the Vitelline and allantoic circulation, which are the sources of chorionic vascularization, there exist essentially two basic types of placenta. The first one is choriovitilin placenta or Yorkshire placenta, and the second one is chorioallantoic placenta. In choriovitilin placenta, the allantois remains relatively small and never makes contact with the chorion. The yolk sac, on the other hand, becomes very large and fuses with the chorion. In these mammals, the chorion receives its blood supply from the yolk sac or vitelline circulation, and the placenta is thus called choriovitilin placenta. However, in marsupials, although only a portion of the yolk sac and thus the chorion is provided with vascular mesoderm, it is still referred to as yolk sac placenta. In chorioallantoic placenta, for example, in case of Euthian mammals and in some marsupials such as Paramelis and Deciurus, the yolk sac remains rudimentary while the allantois becomes well developed and it fuses with the chorion and provides the chorionic circulation. This type of placenta is called chorioallantoic placenta. From this diagram, we can very easily find out the basic difference between choriovitilin placenta and chorioallantoic placenta. In choriovitilin placenta, the yolk sac is very well developed, whereas the allantois is poorly developed and it is rudimentary. Examples are the didelphys and macrophage. Didelphys is a opossum species found in Argentina, Bolivia, Brazil, Paraguay, and Uruguay, and Macrophage is a marsupial species found in eastern third of Australia. Chorioallantoic placenta is opposite to the choriovitilin placenta, where the yolk sac is poorly developed or it is rudimentary, and allantois is well developed. Examples are the Paramelis and Deciurus. Paramelis is the smallest bandicoot formerly occurred throughout the southwest of Western Australia, and Deciurus or eastern quail is a medium-sized carnivorous deciurid marsupial which is native to Australia. Now come to the other basis of classification of the placenta. Placenta may be classified based on the contact between chorionic villi and the endometrium. Based on this, they may be of three different types: non-deciduate placenta, deciduate placenta, and contradeciduate placenta. However, placenta may be classified based on the distribution pattern of chorionic villi in different mammals, and the pattern of distribution of the villi varies from species to species, and accordingly it may be classified into diffuse placenta, cotyledonary placenta, zonary placenta, and discoidal placenta. Discoidal placenta may be monodiscoidal and bidiscoidal placenta. Again, five different types of placenta are recognized according to the histologic relationship established between the chorion and the uterine wall. It is generally considered one of the most useful and instructive methods for functionally describing placental types, and it was proposed by Grosser. These five types are epitheliochorial placenta, syndesmochorial placenta, endotheliochorial placenta, hemochorial placenta, and hemoendothelial placenta. The hemoendothelial placenta again classified into hemomonochorial placenta, hemodichorial placenta, and last one is the hemotrichorial placenta. Now, on the basis of closeness of fetal and maternal tissues, placenta may be of the following three types. They are non-deciduate placenta, deciduate placenta, and contradeciduate placenta. In non-deciduate type of placenta, the chorionic villi lie on the crypts of uterine wall and do not fuse with it. At the time of birth, chorionic villi are simply drawn out from the depression without any damage to the uterine wall. Therefore, no bleeding occurs. Examples are the pig, cattle, horse, and other ruminants. In case of deciduate type of placenta, the allantochorionic villi penetrate deeper into the uterine tissue. At the time of birth, the fetus with its membranes, including the chorion, is removed. As a result, the uterine lining tears away with extensive hemorrhage or bleeding. Contradeciduate type of placenta are found in Talpa and Paramelis. No part of the placenta is thrown out in such cases, and the fetal portion of the placenta remains in the uterus, both of which is absorbed in situ by maternal leukocyte. Now let me define each and every type of placenta according to their classification. In diffuse type of placenta, the chorionic villi remain scattered all over the chorion. Examples are the lemur, pig, and horse. In cotyledonary placenta, the true villi are found in groups or patches, and these groups or patches are called cotyledons. This type of placenta are found in cattle, sheep, and deer. In zonaric placenta, the chorionic villi are developed in the form of a belt or girdle-like band around the middle of the blastocyst or chorionic sac. Examples are the cat, dog, etc. Discoidal type of placenta are found in insectivores, man, bat, mouse, rat, rabbit, etc., where the villi are restricted to a circular disc or plate on the dorsal surface of the blastocyst. Now the epitheliochorial placenta. This type of placenta is the most primitive and most superficial placenta, and it lacks significant invasion of the uterine lining. No destruction or invasion of the maternal tissues occurs, and no layers are therefore removed. Six tissues lie as barriers between the fetal and maternal blood. They are endothelium of maternal blood vessel, endometrial connective tissue, uterine epithelium, chorionic epithelium, allantochorionic connective tissue, and endothelium of fetal blood vessel. In syndesmochorial type of placenta, only five barriers lie between the fetal and the maternal bloodstream. This type of placenta are found in cattle, sheep, etc. In this type of placenta, the fetal and maternal components are fused so intimately, which results in the destruction of the uterine epithelium and thus bringing the chorion into contact with the connective tissue of the uterine mucosa. Whereas in endothelial placenta, only four barriers lie between the fetal and maternal bloodstream. This type of placenta are found in carnivores such as dog, cats, bears, etc. Here, the uterine mucosa is also reduced and the chorionic epithelium comes into contact with the endothelial wall of the maternal blood vessels. Hemochorial type of placenta is the most invasive placenta. Here, all maternal tissue layers disappear through erosion, which leads to direct connection between the chorion and maternal blood. Here, only three barriers lie between the fetal and maternal bloodstream. They may be classified again into hemomonochorial type of placenta found in primates, hemodichorial type of placenta found in rabbits, and the hemotrichorial placenta which are found in rats and mice according to 1, 2, and 3 trophoblast layers respectively. In hemomonochorial type of placenta, there is a single layer of syncytial trophoblast. In case of the homodichorial type of placenta, there is a single layer of syncytiotrophoblast and a cytotropoblastic layer. And in case of the hemotrichorial placenta, there is a single layer of cytotrophoblast layer and two layers of syncytiotrophoblast. This table shows a gradual reduction of placental barriers between fetal and maternal bloodstreams. In epitheliochorial placenta, there exist three fetal layers and three uterine layers. Therefore, it is 6. In syndesmochorial placenta, there exist three fetal layers and two uterine layers. Therefore, it is 5. In endotheliocorial placenta, there exist three fetal layers and a single uterine layer. Therefore, it is 4. In hemochorial placenta, there exist three fetal layers, no uterine layer. Therefore, it is 4. And in hemoendothelial placenta, there exist two fetal layer and no uterine layer. Therefore, it is 2. The placental barriers exist between the fetal and the maternal bloodstreams is not the true barriers. The beneficial substances that can cross the placental membrane are the oxygen, carbon dioxide, glucose, L-form amino acids, free fatty acids, vitamins, water, sodium, chloride, potassium, iodine, calcium, phosphate, urea, uric acid, bilirubin, fetal and maternal RBCs, maternal serum proteins, low-density lipoproteins, prolactin, steroid hormones unconjugated, immunoglobulin G and immunoglobulin A. Harmful substances that can cross the placental membrane are the viruses, for example, rubella, variola, measles, X-drugs, which are the absolute contradiction in pregnancy. For example, thalidomide, aminopterin, oral contraceptives, nicotine, alcohol. Category D drugs. D drugs are the definitive evidence of risks to fetus. For example, tetracycline, carbon monoxide, organic mercury, lead, polychlorinated biphenyls, potassium iodide, cocaine, heroin, etc. Substances that do not cross the placental membrane are the maternally derived triglycerides and phospholipids, protein hormones, for example, insulin, drugs such as succinylcholine, heparin, methyl dopa, etc. Immunoglobulin D, immunoglobulin E, immunoglobulin M, and bacteria in general. The placenta is what which nourishes and protects your baby in utero. Most of the time, or in case of a normal placenta, it attaches to the top or side of the uterus. But if it settles lower down, it might result in placenta previa. In partial previa, the placenta covers part of the cervical opening. In marginal previa, the placenta borders the cervix, and in complete previa, the placenta covers the entire cervical opening. In all cases, except normal, the placenta physically blocks the opening to the birth canal or the vagina in some way, which means it could affect how your baby comes into the world when it is time to deliver. Placenta has a crucial role throughout the fetal development. The fetus completely depends on the placenta to survive. Therefore, the placenta is a major lifeline between the mother and the fetus. The functions of placenta are manifold, and this can be discussed in the following way. Anchorage functions of placenta. Placenta serves as adhesion or anchorage of the developing embryo with the uterine wall. Nutritional role. Monosaccharides, lipids, amino acids, vitamins, and hormones pass by diffusion or active transport through the placenta. Respiratory role of placenta. Gaseous exchange takes place by diffusion across the fetal membrane. Excretory role. Waste products like urea, uric acid, and creatinine are eliminated via the placenta from the embryonic blood to the maternal bloodstream by diffusion. Storage functions. Glycogen, fats, and some inorganic salts are stored in the placenta to be utilized when diet of the fetus is inadequate. Enzymatic functions. Placenta produces various enzymes such as diamine oxidase, oxytocinase, phospholipase A2, which protects the fetuses. Endocrine functions of placenta. Placenta acts temporarily as an endocrine organ. It secretes many hormones such as estradiol, progesterone, chorionic gonadotropin in most mammals, and also placental lactogen in human female. Immunological role. Antibodies produced against certain diseases in mother are passed on to the fetus. Destructive function. Pathogenic organisms and drugs used during pregnancy can cross the placenta and cause disastrous effect on the fetus. These are few suggested readings and references which I used to prepare this PowerPoint presentation. I do sincerely hope that after going through this PowerPoint presentation, you will be able to get some ideas and informations about placenta. Here is my email ID. I would appreciate receiving your any queries about this topic. Thank you so much.